Bismillahirrahmanirrahim. Today the topic of discussion is microscopic structure of lymph node and tonsil. Our objective of this lecture will be we will talk about what is lymphoid tissue, what is the location of different lymphoid organs, and specifically we will talk about two lymphatic organs, lymph node and tonsil. So we start with lymphoid tissue. Lymphoid tissue is a type of reticular connective tissue rich in lymphocytes. Mainly it is a type of connective tissue, and this tissue is full of lymphocytes, and these lymphocytes are either they are present in the connective tissue in free form, or they are surrounded by a connective tissue layer which is known as capsules and forming the lymphoid organs. So actually when we say the term lymphoid organs, it means we are talking about a collection of lymphocytes surrounded by connective tissue. Those lymphocytes which are present in the connective tissue in the free form means that they are dispersed in between the connective tissue, which is loose connective tissue, and they don't have any proper covering. As we see this microscopic structure of the lymphoid tissue, generally it appears in dark bluish coloration because we know that the nucleus of the lymphocytes are larger, so also they have less cytoplasm because most of the structure is taken by the nucleus, or space is taken by the nucleus, so cytoplasm is very less. So when we stain it with H&E stain, hemotoxin and eosine, it gives blue color, or appearance of the lymphoid tissue is bluish or purplish. And the main stroma of this lymphoid tissue is formed by reticular fibers. So there is a meshwork or network of reticular fibers which are present inside the lymphoid organs, and they act as a supporting or making an infrastructure of the lymphoid tissue or lymphoid organs. And these reticular fibrils are secreted by fibroblastic reticular cells which are present in the lymphoid organs. The stroma is formed by reticular fibrils, and inside those network we have a lot of lymphocyte present there, forming a lymphoid tissue and lymphoid organ. We can classify lymphatic system or lymphatic tissue in different ways. So if we talk about in terms of the architectural types, it can be diffuse or it can be follicular. So in two types it is appearing. In the diffuse form, that it appears uniform appearance in the connective tissue and scattered, while in the other form it appear in a spherical structure which we name as follicular, and those follicles are actually consist of lymphocytes and they are enclosed by connective tissue. So on the basis of architecture we can say they can be diffuse and they can be follicular, and according to the covering of the connective tissue they can be encapsulated and they can be unencapsulated or partly encapsulated. Encapsulated means that they have a complete connective tissue covering, and the example is spleen, thymus and lymph nodes. So we see their structure, they are covered by capsule, while unencapsulated structures are tonsils, Peyer's patches in the ileum, lymphoid nodules in the gastrointestinal tract, in the respiratory tract, urinary and reproductive tracts, and they are not covered by or completely covered by capsule. That's why they are known as unencapsulated. And specifically these unencapsulated lymphoid tissue is one of the first line of defense against the foreign material or pathogens, because these are the systems: gastrointestinal tract, respiratory tract, urinary and reproductive tract, which are connected to exterior. So they are actually present in first line of defense against the pathogens. If we talk about the type of lymphoid organs, we can divide them into two types: primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs, where lymphoid cells undergo the maturation, that is thymus. Specifically, T lymphocytes are present in the thymus and B lymphocytes in the bone marrow. So initially, the lymphoid organs are formed and get their maturation in thymus and in bone marrow, and later on their function is taken up by secondary lymphoid organs, which are lymph nodes, spleen, Peyer's patches, lymph nodes, duels of JIT, and other symptoms. So these all systems, respiratory, reproductive, and gastrointestinal tract and urinary tract, they contain the functional lymphocytes in them, and they are known as secondary lymphoid organs. So earlier, the primary lymphoid organs are functional at the time of birth and afterwards, and later on in the adulthood, the function of these primary lymphoid organs are taken up by secondary lymphoid organs. This diagram is showing the location of different lymphatic organs present in the body. We start with the tonsil, which is present at the posterior aspect of the oral cavity on the lateral wall. Thymus, which is present at the lower part of the neck, um, just in the upper part of the thorax anteriorly. Then we have spleen, which is present in the left hypochondrium in the abdomen. And then you can see this blue coloration. These are known as Peyer's patches, which are again the aggregation of lymphatic tissue in the intestine. And we have another very important lymphatic organ known as appendix, which is again the part of intestine. Now, when we talk about lymphoid organs, we should know a term which is known as lymphoid follicles. So first we will talk about the lymphoid follicles, and then we will move to the lymphoid organs. So lymphoid follicles are nodules of densely paired lymphocytes located in all peripheral lymphoid tissue, and mostly these lymphoid follicles are containing B lymphocytes. And we see the structure of these lymphoid follicles. We can see that this spherical structure can have two distinct areas: a peripheral darkly stained area, which is known as mantle or peripheral zone, mainly have small and resting lymphocytes, or in other words we can say naive cells, while in the central part we have germinal center, which is light stained or define the secondary or reactive lymphoid follicles having larger and activated B lymphocytes or lymphoblastic cells. So we see the picture of the here we can see this is a lymphoid follicle present inside. So in the center, if we can compare with this portion, this is darkly stained, smaller and darker as compared to the central part where we have a little bit larger size of cells, lymphoblastic cells, along with we have light stained area. So this light and stained area is known as germinal center, and the peripheral area, which is known as ventral or cap or dark stain, is known as peripheral zone. So each lymphoid nodule is generally comprised of two areas: peripheral zone and a central zone. Peripheral is known as mantle and central is known as germinal center. So we will come across this lymphoid follicle in different lymphoid tissues. Here another diagram, we can see this whole is lymphoid tissue. Lymphocytes are scattered all around, but in the center we can appreciate that we have a lymphoid nodule, and this is the central zone and this is the peripheral zone. Now we will start with the first lymphoid organ, that is lymph node. Lymph nodes are present throughout the body along the lymphatic vessels, and they are numerous in axilla, groin, cervical area, and abdomen and thoracic mesenteries. The main function of the lymph node that they filter lymph before it returns to the vasculature. And if we see the structure of the lymph node, formed of two surfaces, one convex surface and other concave. At the concave surface we have an opening which is known as hilum, through which the blood vessels and lymphatic vessels are passing, and also on the convex surface, that is a receiving surface for the afferent lymphatic vessels. Here in this diagram we can see the inguinal as well as axillary cervical lymph nodes. So this diagram is basically showing the presence of lymph nodes in the body. So here these are lymph nodes and they are connected together by the lymphatic vessels, and ultimately all the lymph which is starting from the lower limb, they are going up and ultimately it drain into the venous system just at this level. So we have cervical lymph nodes, axillary lymph nodes, then we have inguinal lymph nodes. These are the three areas where we have cluster of lymph nodes present superficially. Now when we talk about the internal structure of the lymph node, as we have already discussed that it is an encapsulated structure. So a capsule, which is connective tissue, actually is covering the lymph node completely, and from that lymph node there are extension of that inner side of the capsule into the central zone of the lymph node, or inside or internal part of the lymph node, and we name them as trabeculae. So trabeculae are the extension of the inner side of the capsule into the lymph node. And when we talk about the internal structure, we divide the lymph node into two portions, and peripheral outer portion which is known as cortex, and an inner portion which is known as medulla. The cortex contains lymphatic nodules with germinal center, and medulla contains medullary cords and medullary sinus. So here in this schematic diagram we can see that this is the capsule, this brown color structure, and if you can appreciate, from this capsule extensions are going in which we name as trabeculae. So what these trabeculae are doing, they are penetrating inside the tissue. So not only they are supporting the tissue, but they are providing main passage to the blood vessels as well as the lymphatic vessel which can pass through these trabeculae. And if we see the structure, we can see lymphatic nodules are present. We have talked about it has a central zone and a marginal zone. So these lymphatic nodules are present in the peripheral part of the lymphatic lymph node, and we name this area as cortex, and the rest of the central portion we name as medulla. And the junction between the cortex and the medulla, which is not very clear margin structure, we name that area as paracortex. So paracortex means parallel to the cortex. So we can say that paracortex is the junctional area between the cortex and the medulla. Regarding the medullary cords, they are branched cord-like extensions of lymphoid tissue arising from the paracortex, and they are penetrating deep into the medulla. They contain primarily B lymphocytes and plasma cells and macrophages. On the other side, paracortex contains T lymphocytes, and already we discussed that this lymphoid nodule is containing also B lymphocytes mainly. Medullary cords are separated by dilated spaces frequently bridged by reticular cells and fibers. These are known as medullary sinuses. So they are the dilated spaces because they are formed by this continuous endothelial lining, and these spaces are connected to the reticular cells and fibers, and we name those spaces as medullary sinuses. And they contain lymph, the lymphocytes, macrophages, and sometimes granulocytes if the lymph node is draining an infected organ. This is the microscopic structure of the lymph node, and as we have discussed, we can see the capsule of the lymph node. This is the outer capsule, and from the capsule we can see that there are inward extension of the capsule which we named as trabecules going inside. The outer or the peripheral part contains the lymphatic nodules, and that is known as cortex. So this portion is cortex, and on the inner side we have medulla, and in between them the partition area is known as paracortex. Just below the capsule we have a space which is known as subcapsular sinus. From subcapsular sinus these trabeculae extensions are going in or penetrating inside between the lymphatic nodules, and as we have earlier discussed, the lymphatic nodules are stained lightly in the center known as germinal center, and in the periphery they are known as mantle or marginal layer. In the medullary area we have medullary sinus and medullary cords. The lymphocytes in the medulla are arranged in shape of cords. We name them as medullary cords. Now this is a magnified diagram which is showing a lymph node. This is a cortex. This is known as paracortex, and this is known as medulla. Now if we talk about the cells which are present in this lymph node, the abundant cells in the lymph node are lymphocytes, plasma cells, dendritic cells, macrophages, and antigen presenting cells. So lymph node is composed of these all type of cells, and they all are arranged in the stroma, which is formed by the reticular fibrils and reticular cells, especially in the cortical area. So the cortex contains lymphatic nodules, and the medulla contains medullary cords. So these are the collection of lymphocytes, and we name them as medullary cords, and in between them we have spaces which are known as medullary sinuses. And in the paracortex area, this is a very important area because at this place we don't see any lymphatic nodule structure. From here the branching of lymphocytes takes place, and they penetrate like a branching pattern making the medullary cords, and this is the area which is known as paracortex. It is differentiated from the cortical area in terms of that if we go for immunohistochemistry we can see the type of cells. In paracortex the maximum number of lymphocytes which are present, they are T lymphocytes, while when we talk about the cortex, in cortex maximum number of cells which are present or type of lymphocyte, that is B lymphocytes along with the helper T cells. So by this we can differentiate the paracortex and the cortex, that paracortex contains T lymphocytes and cortex contains B lymphocytes mainly, and medulla contains plasma as well as cells, macrophages as well as B and T lymphocytes. Here we can see the capsule, and here we can see the subcapsular sinus. This is a much larger view which is showing the capsule of the lymph node, and from the capsule we can see that the trabecule is penetrating, and just below the capsule we can see the subcapsular sinus. From the subcapsular sinus from the trabecular portion, there are extensions of sinuses which we name as cortical sinuses or trabecular sinuses, which branch internally among the lymphatic nodules. These sinuses are lined by very thin discontinuous endothelium, and that is penetrated by the reticular fibrils. And lymph node, as we say, the cortical lymph nodes, they contain two portions: central, germinal center, and peripheral zone. And if we go to next slide, and we can see here that this is the capsule, this is the trabeculae, and this is subcapsular sinus. So if you see, from subcapsular sinus the space is penetrating as cortical or trabecular sinus in between the lymphatic nodules internally, and these sinuses are reaching till the medullary area. Now this lymphatic nodules contains in the center, known as germinal center lymphatic nodule. Mainly here we have B lymphoblastic cells present, which are rapidly proliferating cells as compared to the marginal cells, which are basically resting or naive cells and have more concentrated and give darker coloration. The paracortex area, as we have discussed, that this is the area, this area contains a very specialized type of postcapillary venules, and we name those postcapillary venules as high endothelial venules. And this high endothelial venule is one of the very important area, because 90% of the lymphocytes are entering into the lymph node through these highly endothelial venules. So this is one of the entry point for more than 90% of lymphocytes into the lymph node. And below that we can see, again we have discussed that the lymphocytes are present in a branching way, and we name them as medullary cords containing B lymphocytes and also containing T lymphocytes, and in between them we have spaces. These spaces are known as medullary sinuses, and these spaces are again formed by discontinuous endothelial lining, and also they contain macrophages with them. If the lymph which is draining into that lymph node is coming from an infected area, we can see different microorganisms present inside as well as the cytokines present there. Now this diagram is showing the capsule, the trabeculae, subcapsular sinus, cortical or trabecular sinuses are present there, and we can see a portion of lymphatic nodule with a central germinal center, where we can see a little bit larger medium-sized lymphocytes or lymphoblasts are under mitosis, and on the periphery we can see densely packed small size lymphocytes along with macrophages and endothelial cells. Now this diagram is mainly showing the flow of lymph, that how the lymph is flowing from a lymph node. So from already we discussed that from the afferent lymphatic vessels lymph is coming, and we can see the valves present inside. So these valves are preventing the regurgitation of the lymph or backward flow of the lymph, and from the efferent lymphatic vessels the lymph enter into the subcapsular sinus, and from subcapsular sinus through the trabecular sinus or cortical sinus they penetrate inside and reach to the medullary sinuses, which is the final destination for the filtration of lymph. And from this medullary sinus the filtered lymph enter into the efferent lymphatic vessel and go out from the concave surface. And here we can see the macrophages, T cells and endothelial cells present in the medullary sinus. So the flow is from afferent lymphatic vessel, then subcapsular sinus, then trabeculae or cortical sinuses, and then reach to medullary sinuses, and from medullary sinuses it enter into the efferent lymphatic vessel. Uh, the next is palatine tonsil. So tonsils are actually encapsulated, unencapsulated structures that they are not completely covered by capsule, and they are present in the pathway of gastrointestinal tract. So in the next lecture we will talk about more about their presence or in which group they are present. Here we will talk about that the surface of the tonsil is covered by stratified squamous non-keratinized epithelium which is facing towards the oral cavity. As we have discussed, the tonsil is present in the oral cavity posteriorly on its lateral wall. So the part of the tonsil which is facing towards the oral cavity, it is lined by stratified squamous non-keratinized epithelium which is the epithelium of the oral cavity, and the surface is not smoother of that area, but it is having multiple grooves. 10 to 20 grooves are present, and those grooves are known as tonsillar crypts. And below epithelium we have lymphatic nodules present in the connective tissue, and dense connective tissue underlies the palatine tonsil and forms its capsule. So we can see the structure. Here we can see this is a diagram which is showing a palatine tonsil. This is the base on which the tonsil is resting. So they are skeletal muscles of the oral cavity. Just above them we have a capsule. So that capsule is not completely enclosing it. It's just only present on the base. And from the base this capsule is extending inward, or we can say extending outward or deeper into the tonsil, and we name them as trabeculae. And if we go through the surface we can see the lining epithelium of the tonsil are known as stratified squamous non-keratinized epithelium, and the surface of that is not smoother, it has multiple grooves, and these grooves are known as tonsillar crypts. So why these grooves are formed? Because as we have already discussed in microvilli, the main function of microvilli is to increase the surface area. The same these crypts are doing with it, that they are increasing the surface area. So in a small place many lymphatic nodules are adjusted. So just below the stratified squamous non-keratinized epithelium we have lymphatic nodules. So there is a cluster of lymphatic nodules present just below them. So we have a base, we're having a capsule. From capsule trabeculae extending up. These trabeculae extending up, they contain lymph vessels. Along with that they also contain capillary and venules. And on the outer surface we have, or the oral surface we have stratified squamous epithelium, and below that we have lymphatic nodules, and this surface is deeply grooved by crypts which are known as tonsillar crypts. So this is all about the palatine tonsil. So thank you very much. If you have any question you can ask on my email address. Thank you once again.